So in lecture, we just finished looking at that last step of cellular respiration, the electron transport chain, and I mentioned that the electrons do have to go somewhere. So they have lots of energy in them, they can pump hydrogen ions across, but they do eventually have to go somewhere, so they have to find what's known as the final electron acceptor. And for a lot of organisms, aerobic organisms, if oxygen is present, those final electrons, where they're going to end up is tying themselves to oxygen, which in turn usually carries a couple hydrogen ions as well with it. And this is why we get a lot of water as our final outcome in aerobic respiration. So in the electron transport, again, they have to find that final electron acceptor. Aerobic respiration, they tie it to oxygen. It's called inorganic because there's no carbon associated to it. It's just an oxygen molecule all by itself. And when organisms can do this whole process aerobically, if oxygen is present, it is where we get our highest yield of ATP. That organisms could get somewhere between 38 to 40, depending on the organism, 38 to 40 molecules of ATP per glucose molecule. Now, if anaerobic respiration takes place, meaning some of these processes happen when oxygen is not present, so it can't be a final electron acceptor, they have to find an inorganic molecule. Anything other than oxygen. So we're looking for some type of molecule that does not have the carbon attached to it, hence it's inorganic and it's not oxygen. And a lot of times that molecule is carbon dioxide. And this is usually when students are like, but that has carbon in it. Yes, there's always an exception to every rule. Carbon dioxide is technically organic, but because it's got a carbon, but we generally discuss it and prioritize it as inorganic. And that's just because it's not a molecule in itself that is a building block of something. Like glucose, getting broken down for energy, contains carbon. This carbon dioxide is in the air. It's not a building material itself, which is why we classify it as inorganic. So it's a lot rarer for anaerobic respiration to take place just because it has to be a specific inorganic molecule. So some type of prokaryotic organism that can do all of these processes without oxygen. And then in fermentation, which we're going to get into next, there again still has to be a final electron acceptor, and that's when we finally have an organic molecule, a carbon-based molecule like sugar, which is C6H12O6, lots of sugar in that, or lots of carbon in that, and we could find that as a final electron acceptor in fermentation. So in fermentation, the cells require a constant supply of NAD+, and I'm going to go back and forth between these two images. So the very first step of fermentation is still the glycolysis step, where we still started with the 6 carbon molecule and we end up with 2 molecules that are 3 carbon pyruvic acid, or pyruvate depending on your book. So still the first step, and it's only what happens to that pyruvic acid after the glycolysis step that really leads into what we more consider the fermentation part, just because glycolysis is part of both steps. Now my note, as a little recall, in this glycolysis step, we did put 1 glucose of molecule in, we put some ATP to get everything started off, we got a little ADP out, we got some NADH out, and to get NADH out, we did actually have to put in some NAD+. That's an NAD+ means it just doesn't have that electron carrier yet. It doesn't have that, it hasn't picked it up yet. It's only when it becomes NADH or it's picked up that electron carrier. It's picked up the electron. So, fermentation, we are going to take that pyruvic acid, it's going to go through more steps, and depending on the organism, depends on what that pyruvic acid is going to turn into. So, this rest of fermentation, it's going to take NADH, the electron carriers, and it's going to take that energy and the electrons and generate more molecules. This is also where we get some NAD+, meaning is we have this molecule that doesn't have an electron, and it's these NAD pluses that actually go back into glycolysis to be able to pick up electrons from the breakdown of glucose. So it does end up a little bit of a cycle of this part of fermentation makes the NAD+, which then goes to the cellular respiration to pick up more electrons to make more NADH, which then goes right back in here, and it kind of goes back and forth between the NADH and the NAD+. So fermentation provides that source of NAD+, those electron carriers that don't have an electron yet. And fermentation itself is the partial breakdown of sugar. So originally started off as glucose, we made the pyruvic acid, and it starts to break down into other molecules. It can release energy. It's gonna eventually find some type of organic carbon-based molecule as its final electron acceptor. And so what do we actually get out of it? Well, we get carbon dioxide out of it, and we get some other organic carbon-based products. Now the net energy, the only thing we get out of fermentation is 2 ATP. And those 2 ATP actually come from the glycolysis step, where we put 2 in, got 4 out, so we netted 2. Because glycolysis is part of fermentation, we get 2 ATP out, but there's no other ATP coming out from any further breakdown of the pyruvic acid. So I said before, we do get some carbon dioxide out, and we get some other end products. Couple examples of end products, lactic acid. This is what our cells make, our muscle cells. When they get worked too hard, they go into an anaerobic state. And so instead of doing cellular respiration, our cells start to do fermentation, and they make lactic acid, which makes you sore the next day. Other organisms can take pyruvic acid and turn it into things called ethanol. This would be some of your drinking types of alcohol. Our cells don't do that, otherwise you'd get drunk if you worked out, which would be entertaining, and maybe more people would work out, not sure. But different organisms make different end products out of the pyruvic acid. This is showing some of the examples of that. So these are different organisms. Again, they're going to go through glycolysis. They're going to take glucose, make pyruvic acid. This is where you get some of those electron carriers. And then it gets further broken down. And that's where we take that electron carrier, use the energy in the electron, and pump NAD plus right back to the beginning. But some of the things we can get out. Well, if you're a bacteria called Probionic Bacteria, you are responsible for making Swiss cheese. That carbon dioxide is what causes the bubbles, the holes in the Swiss cheese. Those are truly just carbon dioxide pockets. And that acid ending is what gives it kind of a sharp flavor to it. Other organisms other than ourselves can make lactic acid. Aspergillus, Lactobacillus, Streptococcus can all take pyruvic acid and make lactic acid, which gives that sharpness to cheddar cheese, to yogurt, and to soy sauce. Saccharomyces can take pyruvic acid and make ethanol, as well as carbon dioxide is a byproduct. This is a yeast. And so in the beer and winemaking process, there is a ton of fermentation and a lot of carbon dioxide that's given off during that process. And Clostridium, the same genus of bacteria where we get C. diff, can actually, different species can take pyruvic acid and make acetone in your nail polish remover, or isopropyl alcohol, your rubbing alcohol. So different organisms can make different end products from pyruvic acid through their fermentation steps. Kind of cool. Now, we did talk a lot about catabolic pathways and breaking things down, specifically breaking down that glucose or carbohydrates, but there is a whole reverse of an anabolic pathway. And an anabolic pathway means we are making things, so these are synthesis or making reactions that require energy and they require precursor molecules to build larger molecules. So we're just taking some of these smaller molecules, taking some of the energy we've generated, and we are building larger molecules. Where does all the energy and the ATP come from? Well, it comes from the catabolic processes. As we break down glucose, we got a ton of ATP out of each glucose molecule, 38-ish, depending on your organism. And that ATP then is used in our anabolic pathways. Now, most anabolic pathways are reverse of the catabolic pathway. Meaning, an anabolic pathway, we can take a small molecule A, a small molecule B, and we can put it together. We're going to make a larger molecule called AB. So we take two small individual molecules and make one larger molecule. That's building things up. But it can be reverse. We can take one large molecule called AB and break it down into two smaller molecules in a catabolic pathway. There is a specific enzyme that would run each reaction. One enzyme is used for building, one enzyme would be used for breaking things down. But reverse all the time. We are constantly breaking things down and building things up. Now, I call this, the integration and regulation of metabolic function. How our cells like to be lazy. So this is how they're trying to be as efficient as possible or lazy as possible. So cells are only going to synthesize or make enzymes when the substrate is available. If you are an enzyme and you're trying to break down fructose, why would you make enzymes if no fructose is available? It's like making an enzyme that we know is not going to do anything. So cells are not going to make enzymes unless the substrate is there to do something with it. Cells are going to catabolize or break down the more energy efficient choice if two energy sources are available. So glucose is a very simple sugar, and it generally is going to give you the most ATP for, the process of breaking it down compared to other molecules, other proteins, other sugars. And so they're going to make the one that's going to give us the most bang for our buck. They're going to break down. Cells are going to synthesize or make metabolites, various molecules that they need, and then they're going to stop making them if that metabolite is available. It's like we're going to make what we need, and then when we have it, we're going to stop making it. Why make any more? And that feedback inhibition can stop making that molecule when a lot of product is in abundance. I showed that as that pathway where the red circle goes in and makes the yellow triangle, and then we've got the squares, and eventually makes the end product of the green ovals. Well, when that product, the green oval, it is in abundance, that green oval then becomes an inhibitor and stops the whole process. Again, it's let's make things we need, and then let's stop making them when we already have them. It's how our cells like to be lazy or more efficient. And then I like to throw this in there just to show that there's a lot of things breaking down and building things up. And although through all of these processes, when we break something down, there is a lot of intermediate molecules that are made in the breakdown of something. But all those intermediate molecules could also be building blocks to making something else. So everything that has a purple arrow is a catabolic pathway. It's the process of breaking things down. Everything with the teal or green arrow is building things up, is we're making things. So this is showing through that glycolysis step. We have glucose, we make pyruvic acid, but there's lots of other things that are happening as it breaks down. And a lot of these intermediate things that we didn't care that you knew about, but a lot of these little intermediate things can also be used in building things right back up. So it's constantly of breaking things down and building things up and breaking things down in catabolism and anabolism. We just specifically focused on the breaking down of glucose or a carbohydrate.